Hey everybody, how you doing? So just having a wee look at a soil sample, because I thought it might give you some artistic inspiration. So yeah, I don't think I'll explain sort of the function of any of the things I'm seeing. I'll try and be a bit more descriptive. Um, so these kind of, unfortunately, you can't see my cursor where I'm pointing. So this is like a fairly decent soil sample in terms of the diversity of the microbiology. It's not the greatest. Um, this, for your information, is times 40, by the way. So times 40, the magnification of what you would see with just your eyes. Um, so here we're just focusing in on a little fungal strand, fungal hyphae. Um, and the focus on my microscope and the camera that I'm filming it on are not on exactly the same plane. So some of it might look out of focus to you, but I try to sort of get a bit of a compromise. This is times 100. So this is a mycelial strand that you can see right in front of you, the thing that looks like a bit like a root. All these little tiny little dots are a mixture of minerals, sand, salts and clays, and a large amount of bacteria. So once we zoom into like 400 times, you'll see all the bacteria moving around. Um, they actually move around because of a sort of phenomenon called Brownian motion, which is something to do with the thermal properties of atoms, and I can't really explain it in any more detail than that. But it's not through their own locomotion that they're moving. They kind of glide and slide very, very, very slowly. So you'd need to have a time lapse to see the true movement of bacteria. So yeah, this is times 100. This fungal strand in front of you looks to be a fairly beneficial one. We've got a certain sort of number of characteristics that we like to define either beneficial or pathogenic species of microbiology into. So one of those, for example, is the cross walls that you can see, called septa in fungi. So once we zoom in here, you can see the little divisions in between the individual cells. So there we go. So you can see that little septa there, all those septa running down the flank of running down the middle of the fungi. So each one of those divides represents a single fungal cell, basically. So once it's grown the full length of a cell, it'll lay down this sort of recalcitrant septa. We can also see little clamp connections, and that's where it passes its DNA basically from one cell to the next, because the septa is actually blocking the movement of nutrients and DNA. So it passes its cytoplasm through this little clamp connection, those little sort of little bauble things that you see on the side of it there. So all these things that are gyrating around, they're all bacteria. We can see that there's not a huge variation in the shapes. We're looking for sort of for about 14 different shapes, which gives us a sort of representation of the diversity of species of bacteria. There's of course millions of species of bacteria, but 14 gives you some sort of good indicator as to variation. So we can see little singular dots that just look like full stops, and they're cocci. Anything that is rod-shaped is a bacillus. This thing right in the very center, this is a fungal spore, the thing that looks like a lemon. So I think I should get pretty nicely in focus there. Yeah, there you go. So I don't believe that's a mycorrhizal, a spore from a mycorrhizal fungi, i.e. the fungi that form an intimate connection with the plant's roots, be that endo or ecto mycorrhizal fungi. But it's probably a beneficial one. The way that you tell a beneficial spore or a VAM spore, or arbuscular forming mycorrhizal spore, from a normal one is that it has a little phosphorus deposit that you can see, which that one didn't. It appears as a little black dot if it's present. So lots and lots of bacteria you can see, very bacterially dominated. And the interesting thing about this, I think I catch one in a moment, is there's a spirochete goes zooming across the screen. And this is because this sample's actually been given to me and held in a airtight jar. So this compost, basically, which is what it is, has gone anaerobic, which means that there's this turnover from the obligate aerobes to the obligate anaerobes. So I think it comes in from the top right to the bottom left at some point, but I think I paused to see it. It's very, very fast moving. And that's a pathogen. So it's a pathogen for humans, it's a pathogen for plants. There's obviously many species of spirilla and spirochetes, but they can be quite nasty, but only in a large quantities. This is seeing a singular individual is not really anything to worry about particularly. These little globulates, I find one right at the end, which is quite nice, are aggregates. And these are all the bits of organic matter, minerals, everything that, all the geology basically, combined with the organic matter of the soil, has been pulled together by microorganisms using their glue, their glomalins, and their glycochromalax. I would struggle with the pronunciation of that one. And these glues are produced by bacteria and by fungi, and it's what gives soil its structure. So this little thing here is a cyst of some description. I'm not sure which species it is, but it'll be a cyst of a fungal there's a little thing zooming across there. That's actually a ciliate, which is a facultative anaerobe, so it can tolerate low or high oxygen conditions, and it thrives in fluctuating oxygen conditions, because it can produce the enzymes required to break down organic matter in both situations. That is the antennae, possibly, of a arthropod, a macroarthropod. For your information, like a sort of single bacterium is about 1 micrometer across, which is 1,000th of a millimeter. So this thing right in the center here, this is a classic fulvic acid. You can tell it's a fulvic rather than a humic acid because of its color. There's many different types of fulvic and humic acids, but you can just group them sort of generically. So this is an aggregate, and the reason the soil takes on the color that it does is because of aggregation. And these aggregates are formed not only by the pulling together of food resources, but they're often sort of secondary metabolites, basically, as a result of digestion from other organisms. That's another hyphae strand there that's broken off. Looks to be a really beneficial one. It's got a nice dark colour to it. Another aggregate there. We've got another fungal strand on the right hand side, which looks good. Yeah. We've got nice cross walls. It's clear and transparent, not so brilliant. It's also got a very narrow diameter, which is again an indicator of something that's not quite as beneficial. If you're talking about plants anyway, growing plants. And I think I do some shadowing here at one point to give you a better sort of view of all the bacteria. So yeah, I'm trying not to talk about their function because I'll talk about that more when we have an actual meeting. Yeah, the camera's not doing very well when it comes to lowering the amount of light that comes through the iris. So that's a beautiful little one right there, and I believe that's a fungal spore embedded into it actually. And then I think it's that thing that's in the north is also a fungi which is reaching out to. It's got its sights set on this aggregate here, which it will be